Dear audience, along with studying theoretical material, the ability of a student to correctly diagnose histological specimens, identify their microscopic elements, accurately describe the features of their structure and function is a mandatory condition for successfully mastering the course of medical histology. The development of these important diagnostic skills related to the topic, muscle tissues, is the focus of this lecture. Muscle tissues is a group of tissues providing for body locomotion and movements of its parts due to a presence of specialized intracellular contractile structures. The muscle tissue locomotive function is structurally based on a universal mechanical and chemical contractile system, which is constituted by a number of fibrillar structures, myofilaments consisting of such proteins as actin and myosin. Classification of muscle tissues is based on the optical properties of their contractile structures when studied in a light microscope. Striated muscle tissues contain specialized organelles called myofibrils, which are transversely striated due to the regular alternation of light and dark bands corresponding to the localization of contractile proteins, actin and myosin. In smooth muscle tissues, contractile filaments are not arranged in specialized myofibrils, and there is no striation. There, they are able to contract slowly, involuntarily, over a long period of time. Striated muscle tissues are classified into two kinds, skeletal and cardiac muscles. Skeletal muscle tissue is formed by muscle fibers, which are multinucleated syncytia, a sort of cell's derivatives, accounting for up to 40% of the human body mass. These fibers are cylindrical structures, highly diverse in length and diameter, with numerous, around several hundred, nuclei located beneath the sarcolemma and striated myofibrils occupying the main part of the fiber. They are capable of powerful and rapid contractions. Depending on their localization in the body, skeletal muscle fibers are divided into skeletal and visceral. Unlike skeletal muscle, cardiac muscle tissue has a cellular structure and consists of cardiac muscle cells or cardiomyocytes connected into a functional network of cardiac muscle fibers. Cardiac muscle tissue is formed by the middle tunic of the heart wall myocardium. There are three types of cardiac muscle cells: typical or contractive, atypical or conducting, and secretory. In smooth muscle tissues, contractile filaments are not organized into specialized myofibrils, hence striation is absent in these cells. This type of tissue is composed of smooth muscle cells. The vast majority of smooth myocytes have a mesenchymal origin and are usually found in the walls of internal organs and blood vessels, thus they are subdivided into visceral and vascular varieties. Only a relatively small part of smooth myocytes has a neural origin. After birth, they form myoneural cells at the iris and ciliary body. Skeletal muscle tissue of tongue. In longitudinal section, the muscle fiber has a cylindrical shape under the outer sheath, sarcolemma. There are numerous elongated nuclei. Myofibrils are located in the center of the fiber. Myofibrils have transverse striation caused by the alternation of dark or anisotropic and light or isotropic bands, denoted, respectively, as A and I bands. The dark and light discs of some myofibrils coincide exactly with similar discs of others, determining the transverse striation of the whole fiber. Between the fibers there are layers of loose fibrous connective tissue, endomysium, and adipose tissue. On a cross-section, muscle fibers have a round or polygonal shape and contain numerous sections of myofibrils in the form of dots. In areas of muscle fibers in the contraction phase, sarcolemma becomes tortuous and areas of spastic contraction appear. They are represented mainly by dark discs. Light discs are not expressed or expressed very weakly. Cardiac muscle tissue, typical or contractive cardiomyocytes. Cardiac striated muscle tissue is represented predominantly by typical or contractive cardiomyocytes connected by intercalated discs into a functional network of cardiac muscle fibers. Between the muscle fibers there are layers of loose connective tissue with blood capillaries. Structures called intercalated discs are unique to cardiac muscle and represent the interfaces between adjacent cells. They contain many junctional complexes, desmosomes and adherens junctions, which together provide strong intercellular adhesion, and gap junctions, which ensure ionic continuity between the cells. Cylindrical cardiomyocytes within the muscle fiber are bounded by intercalated discs, covered with sarcolemma, contain sarcoplasm and nuclei in the central part of the cell. Striated myofibrils are located at the periphery of muscle cells. Cardiomyocytes' nuclei are centrally located. Due to the presence of lateral outgrowths of cardiomyocytes, anastomoses are formed between adjacent muscle fibers. They ensure the transmission of excitation potential from one muscle fiber to another. Compared with iron hematoxylin staining, myofibril striation and intercalation discs are poorly expressed when stained with hematoxylin and eosin. Cardiac striated muscle tissue is represented predominantly by typical or contractive cardiomyocytes connected by intercalated discs into a functional network of cardiac muscle fibers. Cardiomyocytes are cylindrical cells with a centrally located nucleus, connected to each other by intercalated discs. Anastomoses formed by lateral branches of cardiomyocytes provide connections between adjacent muscle fibers. In cross-section, cardiomyocytes have a round or polygonal shape. Their nuclei are centrally located and may not be present in every cell depending on the level of the section. Cardiac muscle tissue, atypical or conducting cardiomyocytes. In the ventricular myocardium, atypical cardiomyocytes form a subendocardial conductive network of cardiac muscle fibers, commonly referred to as Purkinje fibers. Conducting cardiomyocytes are pale-staining cells, larger than the adjacent contractive cardiomyocytes, with sparse peripheral myofibrils and much glycogen. Ultrastructure of striated myofibril, isolated myofibril of ventricular cardiac muscle cell. Myofibrils are specialized organelles of striated muscles, which have a striated structure due to the regular alternation of light and dark bands corresponding to the localization of contractile proteins, actin and myosin, possessing different optical properties. Myofibrils have transverse striation caused by the alternation of dark, or anisotropic, and light, or isotropic, bands, denoted, respectively, as A and I bands. Each isotropic band is separated by a thin dark fibrillar structure, denoted as a telophragma or Z-line. In the middle of the anisotropic band, a light stripe H is found, through the center of which runs a mesophragma, denoted as the M-line. Each I band is formed by thin microfilaments consisting of the protein actin. Each A band is formed by thick microfilaments consisting of the protein myosin. Thin actin filaments of I bands partially extend into A bands into the spaces between thick myosin microfilaments. The structural unit of myofibril is the sarcomere, an area bounded by two Z-lines. According to the sliding filament theory, muscle contraction occurs as a result of the displacement of actin filaments relative to myosin filament without changing the length of either due to the interaction of the proteins that form them. During contraction under the influence of Ca++ ions, there is a mutual displacement of actin and myosin filaments, resulting in a decrease in sarcomere length due to a reduction in the width of isotropic bands, while maintaining the width of anisotropic bands unchanged. Visceral smooth muscle tissue in the wall of vas deferens. Smooth myocytes form complexes of cells tightly adhering to each other. In the walls of tubular organs, they are arranged in layers running in different directions. On a longitudinal section, the smooth myocyte is spindle-shaped, contains an elongated nucleus in the central thickened part and an oxyphilic sarcoplasm. On a cross-section, smooth myocytes are round or polygonal. Depending on the level of section, the cells have different diameters. Their nuclei are not found in every cell. Vascular smooth muscle tissue in the wall of umbilical cord and inferior vena cava. Smooth muscle cells of vascular walls exhibit a typical structure and ensure the required vascular tone. The arterial wall in the cross-section contains numerous elongated smooth muscle cells, located predominantly circularly in the medial sheath, or tunica media. Single smooth myocytes are constantly found in the inner sheath beneath the endothelium. In the large veins of the lower body, smooth myocytes form longitudinal layers, joining in this case into bundles. Neural smooth muscle tissue. Muscle cells of neural origin are situated in stroma of the iris, which is formed by a loose connective tissue with a large number of pigmented cells. The muscle cells constitute the sphincter pupillae and dilator pupillae. Neural muscle cells have the typical structure of smooth myocytes. Their cytoplasm contains actin and myosin filaments, common organelles, and small pigment inclusions. In contrast to smooth myocytes of mesenchymal origin, each myocyte is innervated separately, which provides a high rate of muscle contraction and rapid change of the pupil lumen depending on illumination. Dear students, today we thoroughly examined the features of the histological structure of the main types of muscle tissues in our body. I hope that when studying the microscopic structure of any organ, you will now correctly identify the muscular elements on the slide and describe the peculiarities of their structure and function. Stay with us for our next meetings on the HistoNavigator channel.